Okay, so guys, now what happens is when we talk about cell division can be divided into two main types. Okay, now what are these two main types, guys? We call this thing as your atypical cell division, and the second one we call this thing as typical cell division. Atypical cell division and typical cell division. Okay, but now what is the difference between the two, guys? Now, atypical cell division, this is the one which is going to be seen in your prokaryotes. Guys, this is the one which is going to be observed in prokaryotes. And guys, when we talk about prokaryotes, who are we talking about? We are talking about nothing but we are talking about your bacteria. All the bacteria, okay, the single celled organisms, bacteria, they are going to be out over here. Kingdom Monera, that is going to be atypical. We are going to have your blue green algae, guys. Okay, blue green algae. Okay, blue green algae or your cyanobacteria, guys. Or remember the other name for this, we have cyanobacteria. Or we are also going to have your archaebacteria, guys. These are the primitive types of your bacteria, archaebacteria. All of these, etc., 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 they are going to show you this atypical cell division. Now, guys, what is this atypical cell division? This is nothing but our binary fission, guys. This is nothing but our binary fission. So guys, let's try to understand, if one cell wishes to undergo binary fission or typical cell division, what exactly is going to happen? Let's try to figure this thing out. Okay, so now let's imagine, guys, we have your bacteria. Okay, now the bacterial cell is going to be like this. Now, guys, when I said kingdom Monera, okay, so there are how many kingdoms? There are five kingdoms. Kingdom Monera, then we have kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. Okay, so out of those five kingdoms, the first one is your kingdom Monera. Okay, now, guys, what happens? Let's try to see. See here, now you are going to see that this bacteria is going to have its own DNA. So its DNA, how is its DNA, guys? Its DNA is going to be circular DNA. Okay, always remember, bacterial DNA, primitive DNA is going to be circular DNA. Now, when this bacteria wishes to divide by binary fission, guys, what are you going to see? You are going to see that first thing, that this DNA, whatever that is there, guys, this DNA is now going to be duplicated. So you are going to see duplication of the DNA like this. Okay. Now, the moment you are going to see like this, guys, in the next case, you are going to see a small furrow. A small furrow develops out over there. It is something like this. Okay. Now, guys, now what happens is whenever we have this furrow, you will see that one DNA, okay, one DNA will be separated onto this side, and the other DNA, it will be separated onto this side. This is what is going to happen, guys. Okay. In the next case, guys, the furrow that had developed, that is going to keep on deepening. Okay, the furrow keeps on increasing. That means it keeps on deepening, 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 like that. Okay, and now eventually, guys, what are we seeing? See, we are going to see that, oh, this DNA, they are going to be like separated already. And now in the next case, this is further going to be going down. So in the next case, we are going to see that, oh, okay, chalo. So now the furrow has quite developed like this, almost towards the center, something of this sort. And finally, guys, what are we seeing? We are observing that from one cell, we are going to get two new cells. See, there is only a narrow bridge of cytoplasm which is going to be joining these two. In the next case, that narrow bridge of cytoplasm, that is going to snap off. And as that snaps off, guys, you are going to see that from one cell, how many cells are we going to get, guys? We are going to get two new cells. Okay, but now, guys, my question is, okay, my question to you is, guys, what do we call these cells? Can anybody tell me what are these cells called? Anybody? Does anybody have any clue? Yes. Very nice. Very nice, guys. Very nice. What are these? These are nothing but our daughter cell. Guys, this original one, we call this thing as our parent cell. Okay, and these ones, whatever we have got, guys, these are going to be our daughter cells. These are our daughter cells. Okay. So my question is, okay. So my question, okay, let me just see the participants. Okay. Yes. Okay. So guys, my question is, see, I have this statement. Okay. You have to tell me whether this statement is right or whether this statement is wrong. Saloni, my question is to you. Okay. So I have two daughters. Both are girls. Is this statement right or is this statement technically flawed? What do you think? I have two daughters. Both are girls. Yes, guys, quickly chat and answer. And Saloni, you have to unmute and answer. What do you think, Saloni? Guys, others, you have to chat and answer. Okay, this is a very important statement, guys. Sachi, Saloni, are you there with us? Saloni, Sachi, Vaishnavi. Okay, guys, you need to respond. Arsha Patil. Guys, what do you think? Arsha, Dia Patel, Rahul, Tanishka. Yes. Okay. So most of you are saying that this is a right statement. Okay. Only few are saying that this is false. Okay. So one is Dia and one is Vaishnavi. So my question is to Dia. Dia, can you tell me, is this, if this statement is false, why is this statement false, or why is this not right? You have said it is false. I have two daughters, both are girls. Is this statement right? You are saying no. Can you tell me why? You can unmute, right? One minute, huh? Dia, I'm unmuting you, guys. Dia, can you unmute yourself?
Yes, sir.
Ha, yes. So tell me, Dia.
In bio, technically, a new cell is known as a daughter cell. It's not its gender.
No. One minute. Sorry. I did not. We can't hear you. Can you be a little loud, please?
Hello.
Ha, yes.
Yeah. In biology, when a new cell arises, it
Ha.
is technically known as daughter cell. It's not its gender.
Acha, absolutely right. Whatever you're saying is actually right. Guys, once again, whatever she said is absolutely 100% right. Remember, when we talk about humans, okay, in humans, when I say I have two daughters, both are girls, it is a wrong statement. Why is it a wrong statement? Because when I say it is a daughter, it is inherently understood that daughter means girls only. I don't have to say, oh, I am having a daughter, she's a girl. No, we don't say that way. We just say that, oh, I am having two daughters. Okay. So same way, if I say I am having, I am having sons, both are males. No, we don't say that. The moment you say that, oh, it is a male, I mean, it is a son, you understand that, this is going to be a male, right? But now, in the case of your cells, guys, now keep this thing in mind. Remember, whenever we have new cells being formed from parent cells, remember, we are not talking about whether it is a male cell, whether it is a female cell, whether it is gender neutral cell. We are not talking about all that stuff. What are we saying? We are just saying that this was the parent cell, and from the parent cell, we have got two daughter cells, meaning these are obtained from the parent cell. That is what we are saying, guys. Okay, we are not saying that, we are not saying that, oh, this cell is male, this cell is female. Okay, so guys, understand this. This is a general term. This is not used to determine the sex of the cell. Okay, just keep this thing in mind, guys. Okay, so this is nothing but your mitosis. The mitosis, binary fission. Okay, so guys, keep this thing in mind. So this is how our binary fission had taken place. Now, guys, is binary fission seen only and only in your prokaryotes? The answer is no. Binary fission is also seen in your protista as well. But when we talk about your binary fission, that is your atypical thing, we are especially talking about your prokaryotes. Okay, so understand this thing, guys. Okay, but now let's try to move on to the next thing. Okay, so we have typical. Okay, typical cell division. Now, what is this typical cell division, guys? This typical cell division is actually a characteristic of all your eukaryotes. Right now, who are eukaryotes? Eukaryotes are all the ones who are going to be advanced, like Protista. It is going to be fungi, plantae, and animalia. All of them, they are going to be eukaryotes. But now, guys, okay, so for example, if you're talking about protists, okay, plants, animals, fungi. Okay, these are going to be considered as eukaryotes. But now, guys, in this, we are going to see three different types of your cell division. Okay, so let's try to understand what is this. So the first one we are going to talk about, guys, this is called as a mitosis. Okay, now, guys, what is a mitosis? Okay, a mitosis, guys, this is going to be really, really deadly, and this is not what the cell really wants. But this happens by mistake. Okay, but what is the end result of this? The cell will always die because a mitosis is not a proper division. But what is the characteristic, guys? Here we say a mitosis, this is going to be direct division. Okay, now why direct division, guys? Because in this kind of division, the nuclear membrane remains intact. Okay, the nuclear membrane, it remains intact. Okay, whereas in other cases, when you're mitosis and meiosis, the nuclear membrane, it disappears, and then after the division is done and all, once again it is going to reappear. In this amitosis, it is not the case. Okay, so we will see amitosis, but then just to give you the first differentiating points. Okay, and now, guys, after your amitosis, we are going to have the next two. That is going to be mitosis and meiosis. Okay, but now when we talk about your mitosis, guys, once again, mitosis, what did I tell you? This is going to be indirect division. Okay, what is going to happen in indirect division? And meiosis also, this is also indirect division. Okay, so guys, you need to understand this thing, very, very important from the need point of view. Okay, so indirect division means what, guys? The nuclear membrane, this is going to disappear and then it reappears. Okay, it disappears and then ultimately at the end it is going to reappear. There are types in this fission too. Yes, Vaishnavi, what are you asking? Can you tell me? Vaishnavi, can you frame your question a little bit more properly? Okay, no, no. In your binary fission, we just see only this thing. We are not going to see any kind of division out over here. And generally, when we talk about your binary fission, this is more or less similar to your direct division. However, we are not saying it that way. Why? Because in your bacteria and all, guys, we don't have the nucleus and the nuclear membrane only. So that is why we don't say that, oh, here in binary fission, it is also your direct division or indirect division. Okay, so this is going to be separate because nucleus is not present in a mitosis, and your meiosis, we see the nuclear membrane. Sometimes it is there, sometimes it disappears and reappears. So depending on that, we have divided it into direct and indirect. Okay, all right, fine. Okay, guys, now the next thing that we are going to see is the first part out over here. Now, see, guys, we have already spoken of binary fission. Like, see what has happened out over here. The cell has divided into two. Okay, generally, your bacteria, E. coli, and all, they are going to show you this kind of division. Okay, but now we are going to go into your mitosis, and let's try to see, guys, what is mitosis? How is it going to happen? What really happens out over there? Okay, so guys, remember this thing. This is mitosis is going to be deadly. generally the cell is not going to go for this thing. Okay, no matter what, it will try to avoid this because ultimate result, end result is going to be death only. Okay, sure. Okay, so guys, when we talk about mitosis, who was the first person, the first ever person to talk about this? The first person to talk about this, his name was Remark. Okay, and now, guys, when we talked about mitosis, what did we say? We said that, oh, this is nothing but our direct division. What do we mean by direct division, guys? The nuclear membrane remains intact. Okay, at no point it is going to disappear. Okay. Okay, now guys, what happens? Let's try to see. Okay, so let's say we are having this cell. Okay, now let's assume, okay, it can be any cell. Okay, so I'm just drawing one normal random cell like this. Okay, so let's say we have this cell, and inside this cell we are going to have nucleus. Okay, so let's say this is our proper nucleus. Okay, now what happens is, let's imagine this cell wishes to divide. Once again, what is going to happen, guys? You are going to see a small furrow coming up out over here like this, and this is how it is going to be. Okay, but now, guys, what will happen? See, generally, if the nucleus has to be divided, now let's imagine, guys, this thing, this nucleus, let's say this is 100%. Okay, this is 100%, guys. Now, if from here the cell has to divide into two, initially what should happen? This 100% should be converted into 200%, meaning duplication of the DNA, duplication of your chromosomes, whatever it is, that should happen. And then from here, when the two cells are going to be formed, then each one should get 100%, 100%, right? Then only the cell will work properly. Otherwise, it is going to be a problem, right? But in your mitosis, guys, what happens? Instead of increasing or doubling the amount of DNA, the cell works with only 100% of DNA. Now, what happens? Let's try to see. So, guys, as we see this furrow coming out, you are going to see the furrow is also be going to be seen somewhere. It's going to be stretched in your this thing as well. Okay, so you will see that, oh, one part is a little small, one part of your nucleus is going to be a little large. Okay, let's try to see what happens. Okay, in the next case, guys, once again, we are going to see that, oh, the furrow has developed a bit more. Okay, and now this has started to take the shape of two cells. Okay, in the meanwhile, guys, you are going to see that, oh, the nucleus is also going to be appearing stretched like this. Okay, fine. Chalo. So this is going to happen. Okay. In the next case, guys, the furrow is going to deepen even bit more down. It is going to deepen further. And now you are going to see, guys, now you will be seeing that, oh, okay, it is going to be completely like this. Same way, your nucleus is going to be stretched, and now it is going to be at the verge of separation, guys. Okay, it is going to be at the verge of separation. But in the next case, now what is going to happen, guys? The bridge, the small cytoplasm, okay, the small segment of cytoplasm which is joining the two, it is going to suck, it is going to snap off. But now when this thing snaps off, guys, what are we going to see? We are going to see that, oh, we have one cell like this, we are going to have another cell like this. But now, what is with the nucleus? Guys, you are going to see that one cell receives more amounts of nucleus and the other cell receives less amounts of nucleus, right? So let's imagine this left hand side, it received 75% of the nucleus, and the right hand side received 25% of the nucleus. Now, guys, what do you think? Are both the cells going to survive? No. Why? Because even if you say that, oh, one cell has got 75% of the nucleus, it should survive. But no, what if the essential genes are not present? What if the genes for respiration are not present? What if the genes for carrying out daily activities are not present? Then what is the use of such DNA? Mm-mm-mm. Ultimately, guys, what is going to happen? This kind of thing is going to be deadly, and ultimately this is going to result into the death of the cell. That is what is going to happen. So guys, few things that you need to remember always out over here, that here both the cells, here both the cells receive unequal amount, both the cells receive unequal amounts of DNA. Okay, and because neither, and therefore, okay, you can say therefore neither of the cells, okay, neither cell has complete complement of DNA, neither of the cells have complete complement of DNA, and now this is going to result into structural and functional irregularities. Okay, structural and functional irregularities. And guys, if you are going to have structural and functional irregularities, then definitely you are not going to survive. This is going to be deadly. Okay, meaning it is going to result into the death of the person. But now the guys, the question is, where are we going to see this kind of thing? Where do we see a mitosis? So guys, we are going to see this thing in three main places. First one, you are going to see this in the endosperm of your seeds. Okay, endosperm is what? It is the nutritive tissue. This is the one which is going to provide nourishment to the developing embryo inside the seeds. So that is endosperm. So sometimes it happens, not all the cells, guys. Okay, not all cells, some of the cells will show you. So endosperm. Second thing, you are going to see this in some of your embryonal cells. Okay, when the embryo is dividing. Okay, so embryo cells or embryonal cells, or we can see this thing in your fetal cells as well. Okay, embryonal cells or your fetal cells. And third thing, guys, you are also going to see this happening in the meganucleus, in the meganucleus of paramecium. In the meganucleus of paramecium. Now, what is this paramecium? How is it, guys? Let me just show you. See, paramecium is nothing but a slipper-shaped organism. Okay, so it looks somewhat like this. Okay, and now you are going to see that basically this is a protist. So his body is going to be covered by your cilia. Okay, so these are small cilia, guys. Okay, and now what happens is inside this you are going to see two nuclei. Now guys, paramecium has two nuclei. You are going to see one nuclei, it is going to be like, really really large. Okay, this large nucleus, this is called as your mega nucleus. This is the one which is called as meganucleus. And guys, just like this, we have another one which is going to be really really small. That small one is called as your micronucleus. So meganucleus and the micronucleus. This is how it is going to happen in your mitosis. Guys, just have a look, just see if you are okay with this. If you're okay, then we will move ahead with your mitosis. Okay, so just have a look, guys. Is everybody okay? Understood? Everyone? Okay. 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 Chalo. So, guys, let's try to move ahead. Let's try to see the next one, that is your mitosis. Let's try to see what really goes out over there, how things are going to happen. Okay. Okay, guys. So after your amitosis, we are into your mitosis. So, guys, let's try to see who was the first person to ever talk about your mitosis. Now, who was this guy, guys? His name is Walter Fleming, the first guy to ever talk about mitosis. Now, guys, you know what? This guy, Walter Fleming, you know what was this guy working upon? He was working on the larvae of salamander. Okay, and he was seeing some changes taking place in the larvae of salamander at that time. Sir, how will we know if such cells are present in the embryo? Now, we won't be able to actually see, but ultimately, what will happen is these cells are bound to die. So the other cells which are there, they are eventually going to take over. So nothing to really worry as such. Okay, next question is, under what adverse conditions does the cells in the embryo have to perform a mitosis? Now, there can be many triggering factors, like, the environmental factors, or let's say, there are going to be stress factors. Let's imagine there is no proper amounts of nutrition, there is no proper amounts of, oxygen supply, no proper amounts of your waste removal. In certain cases, what happens then, it is going to trigger of that amitosis as the cell division. Okay, so it is like, हड़बड़ी में division किया, that is a mitosis. It is that way. Okay, so guys, let's come back out over here. Okay, by the way, guys, uh, I had a word with Anuradha Miss. Okay, so she told me that, you guys were saying that, she has started with the same chapter. No, guys, both these chapters are completely different. Okay, only thing is we have a small part, that is the cell theory, and the initial part. Only that part is going to be the same for both of the chapters, because she told me that, kids were saying that, I have already started with this chapter, and she is repeating that part. The answer is no, guys. Okay, that chapter, the initial part is same. Rest, as we move ahead, it is going to be completely different, and our chapter, it is completely way different from what she was teaching. Okay, so keep that in mind. Okay, guys. Okay, so now let's try to see. So, guys, when we talk about your mitosis, what exactly is mitosis? How is that going to happen? So, let me just tell you, guys. So, in mitosis, let's imagine we have one cell. Okay, so let's say we have this cell, and this cell is now going to undergo mitosis. And by mitosis, guys, what is it going to do? It is going to give rise to two new cells, that is, two daughter cells. That's it. That is what is your mitosis. So if I have to define mitosis, guys, what is mitosis? Mitosis is nothing but it is the type of cell division. Okay, it is the type of cell division in which one cell divides to give rise to two new cells. To give rise to two new cells. Okay, and both the cells receive complete complement of the DNA. This is very important. Okay, and both the cells receive complete complement of the DNA. Okay, but now, guys, let's try to understand what exactly is going to happen out over here. Okay, so guys, see, we are diploid organism. Now, what do I mean by diploid? Diploid means we have two sets of chromosomes, one coming from the mother, one coming from the father. Okay, so when I say we are diploid, guys, diploid is represented as 2n. Whereas when we talk about your haploid, haploid is represented as N. Now, where do we see N, guys? This is seen in the case of your sperms, or this is seen in the case of your ovum, because they are going to be produced by meiosis. Okay? But diploid means what? Diploids meaning you are going to have two sets of chromosomes. Okay, two sets of chromosomes, and here you are going to have n, meaning it is only one set. Okay, now guys, where do we see diploid cells? Diploid cells are going to be seen all over your body, except the cells, that is the gametes of the testes and the gametes of the ovaries, where we are going to have sperms and ovum, which are going to be haploid. Okay, so this is going to be present overall inside the body. Okay, so just keep this thing in mind, guys. Okay, so now if we talk about like mitosis, guys, let's say mitosis is taking place out over here in your skin cells. So now, guys, what happens is one skin cell divides and gives rise to the same type of your skin cell with the same number of chromosomes, with the same set of chromosomes, guys. So in this kind of division, what you will see, let's say if the parent cell is 2n, if the parent cell is 2n, this is going to divide and it is going to give rise to two new cells. And guys, these two new cells, they are also going to be 2n. They are not going to be nn. Okay. Had they been nn, then it would have meant they were undergoing meiosis. Okay. So that is the different thing. Okay, guys, understood this thing? So once again, okay, what are you going to see? We have two sets of chromosomes. So let's say we have the skin cell. The skin cell, it undergoes division, gives rise to two new cells. These two new cells, guys, once again, they are going to have 2n, 2n only. They are not going to be nn. So guys, let's say if we are talking about the chromosome number. Now, what is a chromosome number, guys? Quickly tell me what is a chromosome number. Quickly, 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 guys. Very nice, Kanaka. Very nice, Darshanita, Tanishka. Very nice. Very nice. Very nice, guys. Okay. What is it? It is going to be 46 chromosomes. Okay. So, guys, let's say we started off with 46 chromosomes. Now, when we are going to divide into two new cells, guys, tell me how many chromosomes will be seen in each of the cells? Quickly, quickly. What do you think? Right, right. Some of you are right, some of you are wrong. But now, let's see. Guys, this is not your, division. See, understand. When the parent cell has 46 chromosome, guys, the daughter cells will also be 46 chromosomes. Keep that in mind. Guys, had it been, now some of you are saying that, oh, it is 23. But see, 23 is what? 23 is haploid, only one set of chromosome. So one set of chromosome will be where? In your sperms or in your ovum. So 23 is wrong answer. Okay. So guys, remember, or let's say instead of your human being, you are, let's say, a dog. Okay. A dog is going to have 78 chromosomes. Okay, a 78 chromosome. Now, guys, when this 78 chromosome divides into two new cells, guys, you tell me each cell is going to receive how many chromosomes on each side? Very nice. Now everybody is giving me the right answer. So each side is going to receive 78 chromosomes. Okay. So guys, get the hang of this thing. Remember, this is not like, your division. Like, one cell is dividing into two. So chromosome number Kachak has divided. No, then it will be meiosis. In mitosis, the chromosome number remains the same as your parent cell. Okay, just keep this thing in mind, guys. Okay, so now because of this, we have few names that we are going to give it to this thing. So guys, when we talk about mitosis, mitosis is also called as equaginal division. Okay, this is also known as equaginal division. Now, why equaginal division? Because both the cells receive equal number of chromosomes. Okay, as both the cells receive equal number of chromosomes. Okay, so this is the first thing. Okay, this is our equivaginal division. Okay, guys, just like this, where did we say you are going to see this thing taking place? See, this is going to take place, oh, throughout your entire body except for your testes and except for your ovaries. So can I say it takes place in all your body cells? Okay, so yes, this takes place in all the body cells. Okay, and guys, body cells, they are also known as somatic cells. Okay, so body cells, other term, we call them as somatic cells. And now, because it is taking place in your somatic cells, guys, what do we say? This is nothing but somatic cell division or somatic division. Okay, so this is another term that is used for your mitosis. Okay, so in all the body cells, guys, except, okay, let me just put down, otherwise you will forget, except your sperms, that is your testes and the ovaries. Okay, okay, testes are going to produce sperm, ovaries are going to produce ovum. Okay. There's another question. Can it be called stem cell division? No, not at all. Why? Because see, stem cell division basically it is like, stem cells basically it is the initial cells which are going to be of the body. Okay. Or we have the bone marrow cells which are going to give rise to other cells. So we cannot generalize this thing because the stem cell basically it is the initial thing. Here when we talk about your mitosis, understand mitosis is going to happen in your body cells, and it is going to give rise to all your same types of cells. When we talk about division taking place in stem cells are also responsible for giving rise to your genitals and the testes and the ovaries. So we cannot call this thing as stem cell division. Okay. So generally, when we talk about stem cells, the idea of stem cells is a little different. So generally, whenever we refer to we are talking about stem cells, the idea is that, oh, these are certain types of cells that can give rise to an entire organism or any organ or any part of the body. That is the idea of stem cell. We generally don't say stem cell division in that case, but stem cell division will show you mitosis. Okay, but we will not call it as stem cell division. Okay, all right, understood. Okay, fine. Okay, but now, guys, let's try to see. Okay, now we need to know a few things. Okay, so guys, see, whenever a cell is going to undergo cell division, okay, there are a few things that we need to know. So, guys, let me just show you a typical animal cell, how it is really going to be like. Okay, so we have this animal cell, which is something like this. Okay, let's say we are going to have our nucleus. Okay, so nucleus is going to have the nucleolus and it is going to have the chromatin fiber like this. But now, guys, right on the top of your nucleus, what are you going to see? You are going to see one dense body like this. Okay, this dense body, this is going to have some radiating rays covering it, and inside it is going to show you one line, one dot, one line, one dot. Now what is this that we are showing, guys? This is called as centrosome. Okay, yes, very nice, guys, centrosome. So let me just zoom in onto the centrosome, guys. So see, we have our centrosome something like this, and we have these radiating rays. Now what are these radiating rays, guys? Can anybody tell me? Yes. Very nice, Kanaka. Very nice, Zui. Very nice, guys. Okay, these radiating rays, guys, these are called as asters. Okay, these are known as asters. And now, inside this, guys, inside this centrosome, what are we going to see? I told you we are going to have one line, one dot, one line, one dot. Now, guys, you know what is this? This is not actually one line, one dot. What is it? These are two perpendicular lines like this. Okay, there are two perpendicular lines which are like this, guys. But now in this angle, you can see it is looking like this. But if I show it to you in this angle, now how is it going to appear? One line, one dot, one line, one dot. Okay, so understand this thing, guys. So this, whatever we are showing inside, guys, okay, this is nothing but, can anybody tell me, guys, what is this? We have the centrosome. Inside the centrosome, what is going to be present? Very nice, guys, very nice. Everybody is giving me the right answer. Very good. This is nothing but your centrioles. Okay, so guys, see, we have zoomed in into your centrosome, and inside the centrosome, we are going to see that one line, one dot things. This is nothing but our centrioles. Okay, Later on, what is going to happen, at the time of cell division, guys, the centrosome is going to split. Yeah, kachak. Okay, at the time of cell division, okay, 1 minute. Uh-oh, 1 minute. Huh. Okay, so guys, at the time of cell division, now we will see what exactly is going to happen. Okay, the same centrosome. Okay, so let's say once again the centrosome was like this. Okay, and we had the, just a moment. Huh. Okay, sure. So now let's say we had this thing. Now, guys, you are going to see that at the time of cell division, this thing is going to split up. Okay, this splits up, and you are going to see that the centrioles, they are going to be separated, and these centrioles, guys, they are now going to be like this. They will be like this. But now, when the separation takes place, guys, even if the centrioles they separate, they will start moving opposite, in the opposite direction of the nucleus. But even if they are moving to the opposite direction, guys, they will be held together with the help of some fibers. What are these fibers? Can anybody tell me? These fibers are now known as spindle fibers. Yes, very nice. These are going to be our spindle fibers. Okay, so this is how it is going to happen. But anyways, guys, as we move ahead, we will talk about this thing. But now, why were we talking about this? Okay, the reason is we need to talk about a few more terms. Okay, so let me just give you a quick idea about it. Okay, guys, 1 minute. I'm just closing this thing because it is showing me some error again and again, so that I'll close it, and once again I'll start the screen sharing process. Acha, by the way, guys, I hope you are understanding whatever we are doing, right? I hope I'm not going fast or anything like that. Yes, guys, quickly chat and answer. Quickly chat and answer. Otherwise, I won't know. Okay. Okay. If at any point you feel that I'm going a little fast or so, then do let me know. Okay. However, I like to take it like really, really slow. Okay. So I generally don't rush. Okay. I take ample amounts of time to make sure that the kids have understood each and every aspect. Okay, and don't be hesitant to ask me any question, whatever it may be. Okay, you may think that, oh, this is a very small question, it is a very silly question. Don't be worried, let me be the judge of it. Okay, I will say whatever question. See, for you it may be like you may think that, oh, this might be a silly question, but no, it can be a big hurdle for you, because your entire understanding may be dependent on that. Okay, so don't ever feel that, oh, my doubt is this much, my doubt is big, or my doubt is small. Let it be. Okay, I will see it and I will explain it out to you. Okay, so don't be worried at all, guys. Ready? Okay. Huh. Okay. Anyways, chalo. So now, guys, let's try to see what is going to happen. So, see, guys, I am showing that this centrosome, it is going to be present in your animals, right? This is your animal cell. Remember, it is absent in your plant cell. It is going to be present in your animal cells only. So, based on that, guys, now we have another few names for your mitosis. Okay, now guys, mitosis in your animal cell and plant cell, okay, they can have a few different names. Okay, now guys, in your animals, okay, in your animals, because animals, it contains your the centrosome is present, right? Because the centrosome is present, therefore, because the centrosome is present, guys, we also call this thing as your centric mitosis. Okay, guys, remember this is also known as centric mitosis. Okay, second thing, guys, I can say asters are also present, right? Asters are also present, so we also call this thing as astral mitosis. Okay, so centrosome is present, asters are present. So because asters are present, we will call this thing as astral mitosis. Okay, so centric mitosis and astral mitosis. But now, guys, when we talk about your plant cell, guys, is centrosome going to be present? No, I just told you centrosome is going to be absent. So because it is absent, we call this thing as acentric. This is also known as acentric mitosis. And second thing, guys, if aster, see, if your centrosome is absent, is the aster going to be present by any chance? No. So in that case, the asters are also going to be absent. So if asters are absent, guys, we call this thing as anastral, anastral mitosis. Okay, so guys, always keep this thing in mind. This is always asked in your, choose the following. Okay, choose the following and all. So that way. Okay, everybody all right with this thing? Sure, no problem. Okay, but now, guys, let's try to go back and let's try to see what is going to be your mitosis, how is it going to be, where do we see this thing. Okay, so let me once again go back to your cell cycle, guys. Okay, and let me just pull this thing. Okay, and let us just try to once again see what was happening out over here. Okay, so guys, see, we had said that, oh, this is going to be the upper part, that is going to be your mitosis, guys. So we are going to be zooming only on that. Okay, we'll be seeing only that part, what is really going to happen out over here. So this arc, okay, we will be talking only of this arc, guys. Okay, so guys, we are talking about this mitosis, this part. Now what am I going to do is I am going to just zoom in onto this arc. Okay, and let's try to see what is going to be actually present inside of it, guys. Okay, so let me just show you. Okay, so this was our entire mitosis. Okay, so let's assume this was that arc that, अरे रे वखड़ा हो गया बहुत. Okay, so let's assume, guys, this was our mitosis. Okay, so guys, when we talk about mitosis, mitosis is actually completed in two main stages. Okay, what are the two main stages? Let's try to see. So guys, in the first case, you are going to say that in first step, you are going to see that the nucleus is going to divide. Okay, so what are we going to have? The first step, the division of nucleus. What do we call this thing? We call this thing as karyokinesis. Okay, karyo meaning nucleus, kinases means division. Karyokinases, division of the nucleus. Okay, so let me just put it down. So karyo, this means nucleus, and kinases means actually division. Okay, so you are going to have division of nucleus. Okay, and now once the division of nucleus is completed, guys, then afterwards we are going to have the second step, that is called as cytokinesis. Now guys, if karyokinesis division of nucleus, can you guys tell me what is cytokinesis? What will it mean? Division of what? Yes, very nice, guys. This is nothing but it is division of your cytoplasm. So, nucleus division, and that is followed by cytoplasm division. Okay, but now, guys, what is this karyokinesis and what is going to happen in your cytokinesis? Let's try to just figure this thing out. Okay. So, guys, in your karyokinesis, guys, remember, karyokinesis is further going to be divided into four more parts. Okay. And now, what are these four more parts or four more stages? Let's try to see. The first one, guys, this is known as prophase. Now, pro means what, guys? Pro means primitive or pro means initial. So, this is the first phase. That is why it is prophase. Second one after this, guys, this is going to be metaphase. So prophase, metaphase. Meta meaning middle. Here something is going to happen in the middle of the cell. That is why metaphase. Third, we have anaphase. Anaphase is actually a small phase. Okay, so we have anaphase. And finally, guys, after your anaphase, we have the last phase, that is going to be our telophase. So prophase, metaphase, anaphase, and telophase. Guys, if a cell has to undergo mitosis, first, after the cell enters from the G2 phase, from the G2 phase, it will enter into mitosis. From mitosis, it will first go into karyokinesis, then prophase, metaphase, anaphase, telophase, cytokinesis. And after cytokinesis, guys, from one cell, we are going to get two new cells. This is how this entire process is going to be like. But guys, we are always going to remember this as PMAT. Let's assume you went to your home, you peed on the mat. PMAT doesn't make sense, but you have to remember it in this fashion. Guys, in your NEET exams, they will give you this question: arrange the following in proper order. They will give you certain options, you have to find out. Okay, so very important thing, guys. You have to remember this by hook or by crook as PMAT only. Okay, but now, guys, once again, if I have to show you the same thing, okay, if I have to show you the same thing in your cell cycle, then what? Now in your exams, guys, in your like, you know, your 11th standard boards, okay, boards meaning your normal this thing, they will ask you to draw the structure of cell cycle. Okay, meaning they will ask you to draw a diagrammatic representation of cell cycle. In that case, guys, you have to show this entire thing. Okay, so how are we going to show it? Let's try to see. So guys, the first thing that we have to see is, see this entire upper arc, whatever that we had shown. Okay, this is mitosis. So I'm going to write this as mitosis on the top. Okay, so I will show it as mitosis. But guys, mitosis, I told you this is going to be divided into two main parts, right? So what are the two main parts? Karyokinesis and cytokinesis. So first one, I will write down here, karyokinesis. Okay, this becomes our karyokinesis. Okay, and now here we are going to put down cytokinesis. 1 minute, I will just cytokinesis. Okay, guys, but now our job is not yet done, because karyokinesis, guys, it is further going to be divided. into four more parts. So after writing karyokinesis, guys, you have to draw a line, something of this sort, and now you have to mention out over here one prophase, metaphase, anaphase, and telophase. So guys, when you will draw the cell cycle, you have to represent it in this fashion only. One minute. Okay, everybody all right with this, guys? Understood this part? Okay, very very important, guys. You have to draw it this way. Okay, henceforth whenever they ask you to draw, you are not going to draw this diagram. You will be drawing only this diagram, guys. Okay, all right with this? Fine. But now let's try to go into your mitosis. Let's try to see what exactly is going to happen. So first of all, guys, everybody is okay until this point. Is everybody okay? Sure, no problem. Okay, chalo. So now, guys, let's try to see what exactly going to happen in your mitosis. Okay, so once again, guys, now remember, before the cell enters into mitosis, guys, the cell is going to go through this entire interface, and then only it is going to enter into your mitosis. So now what happens? Let's just try to see, guys. So first of all, we are going to have, okay, we are going to have the interface cell. So first we need to understand how does the interface cell look like, and then only we can see how it is going to happen in array. Oh, sorry, I missed it. Okay, so first we will see how the cell really looks like in the interface, and then only we will understand, like, what are the changes that are going to happen in your mitosis. Okay, chalo. So first, how does the interface cell look like, guys? Okay, so you are going to see that, oh, the interface cell first, it is going to have the cell membrane like this. The cell membrane is as it is. Then inside the cell membrane, guys, we are going to see that, oh, we are going to have the nuclear membrane. Okay, so we have the nuclear membrane. Inside the nuclear membrane, guys, we are going to have the nucleolus, and inside this we are going to have our chromatin fiber. Okay, and finally, guys, right on the top, what are we going to see? We said that, oh, we are going to have our centrosome. Okay, it's the centrosome with the centrioles. This is how a non-dividing cell is going to look like. Okay, so let me just show all the parts. So this is going to be the centrosome. This is going to be the nucleus or the nuclear membrane. Okay, then we are going to have this as your nucleolus, and finally we are going to have this as your chromatin fiber. Okay, so guys, when the cell is not dividing, okay, when the cell is at complete rest, at that time you are going to see that the cell is going to look something of this sort. But now let's assume that this cell is going to enter into your mitosis. So if it enters into mitosis, guys, what is the phase that it is going to enter into first? It is going to enter, just see, guys, it is going to enter first into karyokinesis. From karyokinesis, it is first going to enter into your prophase. Okay, so the first phase is prophase now. So let's say we have entered into prophase. Okay. But now, guys, the moment we enter into prophase, you are going to see a few changes taking place. Guys, what did I tell you? Prophase. This is going to be the longest phase of your mitosis. And guys, how long is this going to last for? This is going to last for roughly about 50 minutes, approximately 50 minutes. Okay, but now here we are going to see some series of changes taking place. Okay, so what are these changes taking place? Let me just tell you. Okay, so first thing, guys, let's say once again we are going to have our cell something like this. Okay, 1 minute. Let me just quickly copy this thing and duplicate these so we can see some transition taking place. Okay, so guys, in the first case, what is going to happen? Let's try to see. And always remember, see, in the first case, guys, you are going to see the nucleus is going to be as it is. The nucleolus is going to be as it is. Okay, guys, let me just try to change the color. Let me just show you this. This is going to look a little bit more better. Okay, so nucleus is going to be as it is, nucleolus is going to be as it is. There is no change. Let's say inside this we are going to have our chromatin fiber. Chromatin fiber is also going to be as it is. But you know what? The first and the foremost step that is going to happen, guys, is that the centrosome, kachak, centrosome splits, and it is going to give rise to your centrioles. So first thing that we are going to see, guys, the centriole are going to be formed out over here, and now these centriole, the centrosome has split and it has given rise to centrioles. Guys, what did I tell you? What is going to happen to these centrioles? These centrioles, they will try to move towards the opposite pole. See, ultimately this centriole, left centriole has to reach out over here, right centriole has to reach out over here. That is the idea, guys. Okay, so you will see that. Sorry. Okay, chalo. Anyways, okay. So you are going to see that this centriole has to reach out over here. The left centriole has to reach out over there. But now, guys, what happens? Even if we have had these centrioles, they are now still going to be joined together with the help of what? With the help of spindle fibers. Okay. So 1 minute. Okay, these are the ones who are going to be joined together with the help of spindle fibers. Guys, this is the first step that is going to happen. So guys, in the first case, I'll just mention here the centrosome splits. And now the moment the centrosome splits, you are going to get the centrioles. Where are the centrioles going to move? The centrioles, they move towards the opposite poles. They move towards the opposite poles of the nucleus, but then at the same time they are going to be held together with the help of spindle fiber. Okay, they are going to be held together with the help of spindle fibers. That's it. This is the first thing that is going to happen, guys. Okay, but now what is the next step? Okay, the next step, guys, we are going to see that the chromatin fiber that was there, see this, the, you know, the green colored thing that we have shown, that the green colored fiber, that is now going to start undergoing condensation. Okay, let's try to see what is going to happen. So guys, in the next step, okay, now we are in prophase only. Okay guys, we have not changed. Okay, we are in prophase only. Okay, so you still going to see that the nucleus is as it is, the nucleolus is as it is. This, the centrioles, they are going to be moving apart. Still, they are, like, you know, moving apart from one another. Okay, they are trying to reach the opposite poles, once again held together with the help of your spindle fibers. Okay, so let's say it is, the situation is like this only. Okay, but now what happens, guys? You are going to see that this chromatin fiber which was there, now that chromatin fiber, it undergoes condensation. And now as it undergoes condensation, it is going to give rise to this egg-shaped chromosomes. Okay, so this is what is going to happen. It is going to give rise to this egg-shaped chromosomes. Now at this moment, guys, I am showing only four chromosomes. Okay, at this moment I am just showing four chromosomes. Okay, but now let's try to see what exactly is going to really happen. Okay, so guys, here I'll just mention that we are going to have this chromosomes and the chromatin fiber. In the second step, you are going to see that the chromatin fiber, it condenses, and it is going to give rise to your egg-shaped chromosome. A little later, I am going to talk about how is this entire condensation going to really take place. We will talk about that as well. Just a second, guys. I am getting a message from the classes. One moment. Okay, no, the attendance is done, so not an issue. Okay, so now, guys, this is going to give rise to our egg-shaped chromosomes. So the duplication occurs. So the duplication occurs in the second step of prophase, right? No. Duplication will occur in your S phase. Here we are just condensing. I will tell you this thing. Don't confuse yourself, guys. Here we are not doing anything to the DNA. Here we are just showing that the entire chromatin fiber, the thread-like thing, it has condensed to give rise to that X-shaped chromosome. Where is that DNA duplicating? It is duplicating in this S phase, guys. Here the entire change was happening. Okay, yeah, okay. Okay, chal. Okay, so now we are going to have X-shaped chromosomes. That's it. Okay, this is what is going to happen in the next case. Guys, in the next case, what is going to happen? Slowly, you are going to see that the nuclear membrane, it starts disappearing, meaning it starts going away slowly. And now the nucleolus, which is out over here, even that nucleolus, it is going to start disappearing slowly. Okay, so now in the next case, what is going to happen, guys? Here the centrioles, they are still going to reach the opposite poles. They are still reaching the opposite sides. Here you are going to see that they are still going to be held together with the help of your spindle fibers. That thing is going to be as it is. But now suddenly the nuclear membrane and the nucleolus, they start disappearing. The X-shaped chromosomes, they are going to be also out over here. This is what is going to happen, guys. So in the next case, in the third case, you are going to see the nuclear membrane and nucleolus, nuclear membrane and the nucleolus, they start disappearing. Okay, they start disappearing. Okay, and in the last case now, guys, what will happen? The centrioles, okay, they have reached the opposite poles completely. The nuclear membrane and the nucleolus, they have disappeared completely. And now these are going to be held together with the spindle fibers. So the spindle fibers will be like this. Okay, now there are going to be different types of spindle fibers which are going to come, but I'm showing you only the ones which are having the chromosomes. And now you are going to see that these X-shaped chromosomes which were there, they start getting attached to the spindle fibers randomly. Okay, there is no proper order as such. Okay, so they start getting attached randomly. That's it. Okay, so here we are going to see in the last case, guys, Okay, so the nuclear membrane and the nucleolus, they disappear completely. Okay, completely disappear. The chromosomes attach. Okay, now this point is very, very important, guys. Okay, the chromosomes, they attach to the spindle fibers, but randomly. Okay, this is all going to happen in complete random. This is very important because as we move ahead, guys, in your metaphase, this arrangement is now going to be brought to the center of the cell, okay, on the equatorial plane. So that is how it is going to be different. But guys, I still have to tell you what is really going to happen out over here. Like, how is the chromatin going to condense and how is it going to give rise to your X-shaped chromosomes? We still have to see that. But guys, for that, I will take around like, roughly like, 10-15 minutes to explain you that. And we are just about to be done. So what we will do, guys, out over here for the day, we will stop over here. Okay. If you guys have any doubts, anything you wish to ask, okay, just let me know, or else we'll stop over here for the day and we will meet again in the next class where we'll continue with the next phases of the mitosis. Yes. Is everybody okay, guys? Have you guys understood this thing properly, everyone? Sure. Okay, no issues. Okay, so guys, once again, we'll stop over here for the day and I will see you guys in the next class. Till then, take care. Bye bye, everybody. Bye bye. Thank you. Bye bye. Bye. Sir, what is the use of amitosis as it leads to the death of the cell? No, this is not use of amitosis. It is basically a malfunctioning of mitosis. So basically, like, a normal process which was taking place, suddenly, it goes haywire. And then you will see a mitosis. Why have we put it down? Because it does happen. What is important from our point of view? Mitosis and meiosis. But then we have seen that, there is this another cell division also which occurs, which kills the cell. That is why we are putting it down. There is no use as such. Yeah. Okay.